All right, everyone, we're going to go ahead and get started. Thank you so much for being here. My name is Cinnamon Moffett. I'm the Associate Director of Marine Research at OSU, stationed here at Hatfield. And this is a hybrid event, so I wanted to welcome the folks that are in the room, but also welcome all those that are online. Thank you, everybody, for being here. Just wanted to do a couple of logistics. I see some new faces in the audience. Because this is a hybrid event, I ask that folks in the room, if you have any questions, just raise your hand and I'll bring you the mic, and then those folks that are online can hear those questions as well. For folks that are online, if you have any questions, go ahead and put them into the chat box of Zoom. We have a volunteer in the room, Roseanne. Hi, Roseanne. We'll make sure that she answers any of your technical questions or reads out any of your questions from the chat box for today's speaker. But that way we both can interact with the folks in the room and folks online. And again, thank you all for being here. I wanted to just do a really quick announcement for next week, but also for what's happening this weekend. Big Blue Film Festival starts on Friday, so hopefully you have your tickets. If not, go online and you can get the last few are still available, I believe. So this is a big event for us on Friday and Saturday. We have like 155 different films that were submitted from a whole bunch of different countries, all coming here. We have the filmmakers that are going to be on site and are prepping and talking about their films. So it's a pretty exciting event. We're very excited about it. So I hope you can join us for that. I also just wanted to say that these seminar series happen every single Thursday, same place, same time. So our next speaker is on the 30th. We have Madeline Lucas, who's a PhD student from the Department of Earth Space Science from the University of Washington, who will be talking about Cascadia culture. And so it is about a research project about building community resilience through K-12 earth science education. So a very different topic, but that's what makes seminars here fun. We have lots of different speakers talking about lots of different things. Just a reminder that if you want to be involved with any of these, you can go to the HMSC website, go to the events page, scroll down, and you'll see a calendar of events. You can get all the information about when our events start and how to log in. Today's event will also be recorded, so if you'd like to share it with others or see something again, you can go to that same page and see our past recording for all of the events. Again, thank you so much for being here. Today's speaker was invited by Lisa Balance, who is the director of our Marine Mammal Institute here. So I'm actually going to let Lisa introduce today's speaker.
Thank you, Cinnamon, and good afternoon, everyone. It's a real pleasure for me to be able to introduce this afternoon's seminar speaker, Dr. Cornelia Oedekoven. Cornelia comes to us from the University of St. Andrews in Scotland. There, she's a senior research fellow at the Center for Research into Ecological and Environmental Modeling. That I'm not really an insider, but the acronym is CREAM, and it's a famous, very famous institute. She's had a long and productive history there, dating to 2008, when she obtained her Master of Research in Marine Mammal Science. She stayed on to obtain a PhD in statistics in 2013, and she's been conducting research there ever since. Cornelia's projects are broad in terms of focal animals. They include gibbons, birds, and of course, marine mammals. And they're broad in terms of geography. Her study and teaching sites have included places in Germany, Ghana, Kenya, Namibia, Vietnam, South Africa, the United States, and my favorite, the Eastern Tropical Pacific. And I'll tell you a little bit more about that in a minute. The overarching theme that unites Cornelia's work is development and application of quantitative methods for wildlife population assessment, including abundance, how many are there? Survey design, how do we count them? Movement, where do they go? And impact, how do humans affect them? Cornelia and I go way back. Between 1998 and 2018, she spent over 2,000 days at sea as a marine ecologist and oceanographer. And she and I spent a good chunk of those days together in the tropical Pacific, conducting research on abundance and trends of marine mammals and seabirds in a study area that was 1/3 larger than the continent of Africa. And there are a number of us in the audience today who actually were on various research vessel surveys associated with that project. The bycatch and mortality of over 6 million dolphins in the tuna purseine fishery there was a major driver of the passage of the Marine Mammal Protection Act, and it led to the largest research effort on upper trophic level marine vertebrates in the world in terms of years, size of study area, and number of species. Later, Cornelia went on to secure a $1 million award to continue to investigate the impact of that fishery on cryptic calf mortality. And finally, because she won't tell you about it today, I do want to mention that in addition to her science and research expertise, she's an impressive artist. And I'm proud that I have a painting of hers of a spotted dolphin that she created hanging in my office. So with this, Cornelia, let me turn the podium over to you, and we're anxious to hear more about your work.
Can you guys hear me okay? Okay, thank you very much for the warm welcome. Thank you very much, Lisa, for the wonderful introduction, very personal and right to my heart. Thank you for the invitation for me to speak here today and for coming and listen. So as Lisa said, I work for a small department at the University of St. Andrews called CREAM. And what this pretty much includes everybody on the picture. And we work on a variety of topics related to ecological statistics. So we're right on the interface between ecology and statistics. A lot of us are actually from biology that moved into statistics, like myself. But we also have pure biologists, and we have computer scientists, part of our department. Some people work on citizen science data. We work a lot on human impact through wind farms, for example. But today, I want to focus on my research. And as Lisa said, I spend a lot of time at sea as a marine mammal observer or seabird observer or oceanographer. And so those of you who've done this type of work, this is one of the challenges that you face doing field work as a marine biologist. And as a statistician, this is a challenge you face when you do that. So between the two, I'm not sure what's the scarier situation. Both of them kind of invoke the feeling, where's my helicopter to take me out of here? But fears are here to be overcome, and you have to learn to deal with either situation. So I work on other statistics as well, but today I would like to focus on abundance estimation. And David Borges, who's in our department, published a paper where he kind of describes how various methods relate to each other, from mark-recapture, which is a lot of times done for humpback whales, and you can capture and recapture the same animals. So you need to be able to identify the individual animals. And then over to spatially explicit capture, recapture, SECR, where you include information about the location where you captured them. And SECR with additional information, for example, the bearing towards the captured animal. And I'll talk more about that later, so don't worry if you miss a detail here or there. And then out to mark-recapture distance sampling. So I'll be speaking about 3 studies that I've worked on over the past years, and explain what these methods are. But we won't go into too much technical detail. We will focus on the additional information that you need to collect during those studies, you know, instead of just getting counts, but what additional information you need to make your estimate. And So for that, we'll start with just a quick introduction to abundance estimates. So abundance estimates are related to the question, how many are there? So in this case, I'm interested in how many dolphins are in the sea. And then you answer, whichever way you come up with, the answer might be, oh, there were 9, my estimate is 9 dolphins, and my 95% confidence interval are 5 to 14. So how do you go about this? So for example, you can put lines down your study area and survey along those lines. You survey out to some part indicated here by the yellow part, so you know which proportion of the area you surveyed. And if you can assume that you detected every animal in your surveyed area, it's a simple mathematical equation. N is the number of detections you had, little a is the area you surveyed, and big A is the whole area of your study area. So pretty simple math. This type of survey is usually not done for dolphins, but it's done for seabirds, for example, but where you use a very narrow strip. For distance sampling, use something similar but different, where you make detection along those same lines, but you're allowed to miss some animals within the surveyed area. And that comes as the cost at having to estimate another parameter, this little p with a hat on. That's the proportion of, or it can be thought of as the proportion of animals detected within the yellow, within the covered area, or your average detection probability within the covered area. Still fairly simple, but we need to work out how we estimate that p. And that's what I'll focus on in this talk. So imagine a ship going down the transect line and making detections of dolphin schools. And so every dolphin school that has a circle is a detected school. You see some are detected, some are not. And for each detected school, you see a black line, and that's your perpendicular distance from the line to the detected school. That's the additional information you need for distance sampling surveys. And that allows you to estimate your P. We will skip over the technical details as promised, but the important part here to take away is that you need those distances, and the method hinges on the assumption that you have perfect detection directly on the line. That means right on the line where you travel along, and to some short distance out, you need to be sure that you detected every school. If you missed 30% along the line, your bias will be 30%. So how can we get around this? So that's where mark-recapture distance sampling comes in, and that's what we're going into next. For that, you need a second platform that observes the same animals. So for example, you can use acoustics as a second platform. And now you might think, how do you get the perpendicular distance using acoustics? So I'll explain that. So for example, imagine the ship travels along a line, the dolphin makes a sound, and because the sound arrives at the two hydrophones that are told by the ship with some time difference, from that time difference, you can get one angle, right or left. Right or left doesn't matter for distance sampling as long as you get the distance. But so now you have one angle, and then the ship moves a little bit further. The dolphin makes a sound again, and you get a second angle. And then you get two possible locations where the dolphin was, right or left. And you get a perpendicular distance from the line. So this is what we used. This is what it looks like then. The ship and the hydrophone array form the two observation platforms. You make detections now from two platforms. Some dolphin schools are detected by only the visual observers, some are detected by only the acoustics, and some are detected by both. And that's the information you need for mark-recapture distance sampling. Of course, you need to be able to match them. Okay, that detection number 1 made by acoustics is the same as detection number 2 made by the visual team. So you need to be able to match. But on paper, it looks easy, right? So here we have the similar summary. These are the models. There's now a detection model and a mark-recapture model. The important things again here are that you need 2 observation platforms, you need distances from both platforms, and this allows you to estimate your trackline detection probability, which is often referred to G0 in literature, but in my study, I usually refer to it as P0, not to confuse it with G0, G of Y. So we'll refer to it as P0. And this is what my friend Shannon and I and Eric, also from NOAA, we did in this paper that we published a few years ago, where we analyzed survey data, combining the visual detections and the acoustic detections of rough-toothed dolphins. And that was the study year. It's from the Starlight survey. And the important part here is this survey, we only included passing mode survey, and what that means I'll explain in a little bit. But it means a lot to Lisa and Bob already. So anyway, we had quite a few detections between the 2 platforms, and we were able to estimate this P0 for the visual team. And it turned out that that was dependent on Beaufort C-scale, meaning the higher the Beaufort C-scale was, the lower P0 for the visual observer was. And this is in support of a paper that came out in 2015 by Jay Barlow, who kind of had a different way of showing that the observers on the NOAA surveys don't always detect everything on the line, which of course throws you as an observer in a bit of a crisis. But but it's a real issue, and we have to deal with it. So this paper just supports Jay's paper. And then, as Lisa mentioned, Vincent Andrews got the contract to design the next survey for Eastern Tropical Pacific dolphins. And I put a question mark behind improving methods, because this is a study that never fully flourished, because the first stage was making suggestions about the next design and how we can improve things. The second stage, which was also funded, was doing a trial survey where we actually try these things. And then the contractor supplying the drone services, they didn't live up to their expectations or to the contract we had with them, which led to the whole project coming to an end. But this is a project that's really close to my heart, and I wanted to discuss some considerations and ideas that we had here with you guys. So, because it is a great data set, and Lisa said maybe the largest data set for dolphins or for any wildlife assessment. Here you just see on the left abundance estimates with confidence intervals for the years 86 to 2006, and those values feed directly into population models, and including the mortality before the mortality induced by the tuna fisheries from before those surveys. This is the trajectory looks pretty grim, and so there's a strong need to continue those surveys. The last one was in 2006, and these are the completed track lines from the 2006 survey, and what happens at each of those individual lines, you know, is depicted in the plot, which we're now familiar with, what that means. So, and the two red circles are the two parts that we wanted to address with the drone. So we had a long-range drone available for using it as a second platform for a mark-recapture distance sampling survey, and that's what I'm going to go into next. So we also had, so I should say, we were contracted by the Inter-American Tropical Tuna Commission, and the funding was provided by, in a large part, by the Pacific Alliance for Sustainable Tuna, and also the Mexican government. They sponsored the research vessel, which we outfitted with a flying bridge and a drone platform, which you see there in the back. And here's another view of the drone platform. We had a helicopter type drone, which allowed us vertical liftoff and landing while the ship was in full speed. This was the design of the track lines where we were going to go test our methods in 2019. And so this is what it looks like. The idea using long-range drones as a second platform. So now you have a drone flying some distance ahead of the ship, making detections, and then the ship follows, makes detections. And that gives us, again, two platforms which make the observations of the same animal, same schools. But of course, it's never as simple as it looks on paper. So I mentioned passing mode versus closing mode earlier. And passing mode refers to this situation where the ship, oh, I'm going to use pointer here now. Hang on. So the ship starts its survey effort. And when it's over here at the star, it has a detection of a school, which is over here. So slightly ahead. And then during passing mode, the observers just spend a minute or so getting a group size estimate and a species ID, but they never go off effort. So this whole track line is really covered on effort. In comparison, closing mode is when over here, when the ship makes the detection, they go off effort and focus on the school. And get a better idea about school size estimates and species ID. And then once they're done with that, they continue on effort, if it's not too far from the line, in parallel from the line. But that results in that, this part was not surveyed on effort. Okay, so that's the difference between passing and closing mode. And it has some implications for mark-recapture distance sampling. So this is a summary of our considerations. Well, for one, so how do we implement this type of survey? Like, what are down to the details? What are we actually doing with the drone? What is the drone going to do when the ship goes off effort and comes off the track line? Then, in the analysis stage, how do we match drone detections to the flying bridge detections? What challenges do we have here? And then passing versus closing mode, how do we incorporate those off-effort times in mark-recapture distance sampling? So those are all things that need to be considered, and there's not much, if anything, out there in the literature because this is all pretty new. So here's here's some more consideration about the drone flight. So in this situation, we wanted to make sure that we capture the dolphins with the drones before they would react to the ship. So that was one of the reasons, was one of the possible reasons that we might miss some dolphin schools, why P0 is less than 1, because before they enter the visual range of the observers, they might be running away already. So to alleviate that, you need to send the drone far enough ahead to where they're still there, to where they still haven't reacted to the ship. Okay, so and that's one of the things we needed to test, is 5 nautical miles ahead. Is that far enough, or do they react much sooner? Does it need to be 10 nautical miles? So that's one thing to be tested. And also, of course, the drone is much faster than the ship. And so it's not going to just fly straight along the line, but it's going to fly zigzag. We thought about sending it out to 3 nautical miles either side, because that's the distance you include in the abundance estimation as your search strip. So how close do you need to space the zigzags for that to happen? How high does the drone need to fly so it captures a lot of area, but at the same time the ground resolution needs to be good enough to where you can identify the species of dolphins. So those are some of those considerations. And then of course the drone needs to be able to land and start and land without crashing into the ship. So here's an example footage. And I have to say that I mean, I was just less than impressed by the drone company, but the pilot, he was so good. He made it look like child's play. He was like the national champion of 3D helicopter drone flight of Taiwan. And he would always come in and eat gummy bears and then go out and launch the drone, and nothing is a big deal. So he was very impressive. Okay, so in terms of drone flight, what does the drone do when the ship goes off effort? So this is the idea. Let's say the ship has a sighting and the drone is 5 nautical miles ahead. Then we thought maybe it's best then, I mean, it doesn't make any sense for the drone to continue, and then it's like way ahead. But we thought maybe it's best if the drone comes back to where the sighting is and then starts the flight while the ship works the sighting and starts the zigzag flight. So it's again 5 nautical miles ahead. So it's just a little idea. On paper, looks easy, but the implementation is a different thing. So another issue is matching detections between platforms. And let's say a D1 is a dolphin sighting that the drone made. And FB1 is a sighting that the flying bridge made. Now you need to, as the analyst, you need to decide, is that the same sighting or not? And that's not easy at all. So, but then you think about the information you have. You have the location of either, you have the time of the detection for either. You can look at how close that is. Of course, especially the flying bridge location comes with some error, because you always round to the nearest decimal radical. And of course, there's a time gap between the two sightings, so the animals will have moved. But you have additional information, like, of course, the species, the behavior, direction of travel, and so on. And then you can, as a statistician, you get excited about these things, because you can, rather than having it a binary situation, is it a match, yes or no, you can come up with a probabilistic model. Is this a match? I'm 70% certain it is a match, and 30% it's not. So you can incorporate that into your statistical models. Turns out that trying to decide that whether it's a match or not is maybe best done in real time, because you never have as much information right in front of you as when it's actually happening. You compare the the map of the flying bridge has with the map that you have entering the drone sightings. So this is us in action. Juan Carlos is there on the right, of course, and Clarity in the middle, for those of you who know. And so one of the things that the drone company didn't deliver on is the quality of the footage. But I thought I'd show some footage anyway. This is, of course, you know, 5 killer whales. And so we're trying to stay over the school, but you have to imagine the drone is over a nautical mile away from the ship. It's not like right next to the ship where this is happening. And I always use this little footage for my audience to get their computer science hat on. I mean, I assume none of us are computer scientists, but if you were a computer scientist and you had to write an algorithm to detect dolphins in this video footage, what information do you have? So you have, of course, the color difference between the dolphins and the water in any single frame, the picture. You can get the shape of the animal from that. And then, so this is image analysis. And then there's another thing called optical flow. So if you think about from one frame to the next, you have dolphins moving in one direction and the glare and waves moving in another direction. So those are the two types of information that the computer scientists use, that we worked with on this project, to extract the dolphin sightings out of the footage. That's about as much as I know about computer science, so we'll leave it at that. This just sums up what I just said. And here's another piece of footage of some pygmy killer whales, where we tried to get to the true group size of the school. Again, the footage is very poor, but perhaps it shows that there is some value in having video compared to still images, because the animals move in and out of view. But on the other hand, if we had much better footage from a still image, so that is worth a comparison, I think. So I actually went through this and tried to count all the animals in there, and that was one of the easier schools. You're welcome to come up with an estimate. Okay, if we still have time, then we can go into the third study. Or have I been talking for an hour? Okay. Okay, so the next study takes us onto land and into acoustics again, and into a different statistical methods called spatially explicit capture-recapture, or SCCR for short. And we're using angles in addition to just capture histories. Again, we're working here with people from different disciplines. So for example, we have Mark Johnson on the team, who some of you might know. He's the lead guy on the DTAGs, which is used a lot for marine mammals. So this is also his first terrestrial project. And we have computer scientists, and we have biologists in Laos who we collaborate with, and Ben Stevenson and Taryn from New Zealand. They're experts on statistical software development. And this is proof that I'm capable of other field work, although it's pretty easy to spot the marine biologist in there with a big red face. This is in the Annamite Mountains in Laos about a year ago. So again, the question is similar. How many gibbons? Similar type of answer. And I put occupancy in here because gibbon studies are much less developed than marine mammal studies. So a lot of people that use passive acoustic monitoring focus on simply occupancy, which means, are there gibbons present at a site, yes or no? So we want to advance the methods with what we do and come up with a good method for estimating abundance. Actually, abundance of calls, which you then can convert into abundance of animals using some multipliers. So this is what the spectrogram for the northern white-cheeked gibbon looks like. You have some, let's use the pointer. So you have the male calling here, and this is the male again, and then the female starts with a very stereotypical call consisting of a bunch of notes. I always call them upsweep because I do come from the marine world. And then here's the male again. They're territorial, they live in groups, and the dominant male and female, they engage in a territorial song every morning. And this is what you see from one male and one female. And I can try and play. It's cool, no? There's the female starting. So they sing really loud. They sing at about 100 decibels, which means you can hear them well over a kilometer away, up to 2 kilometers, maybe even further, which makes them, you know, really good for passive acoustic monitoring. You know, imagine trying to find one of those gibbons visually in the forest. No chance because they're really shy as well. So we're proposing just to put these recorders out, and yeah, so on the left you see the audio moth, which is just a simple recording device. They're made for use in the water. A different type, you can use them in the water as well, but showing here the one that they use in the forest. And on the right is our recorder that Mark has built for us, where 4 microphones are about 10 centimeters apart each, and that makes it directional. So we can not only determine, you know, if a gibbon was calling, but also the direction to the calling gibbon. And this is how that works. The gibbon makes a sound. It arrives at one microphone sooner than the others. And then you determine the direction to the calling animal. Of course, you don't need to see where it is. You just need to hear it. And you get the direction. Then you put 3 of them at a given site. And the gibbon makes a call. It's detected in this case by 2, but not by the 3rd. So you get your recapture information not detected on the first one on the left, but detected on the other 2. And you get 2 angles to the calling animal. And with that, you get a pretty good information where the animal was that was calling. For spatial capture recapture, you don't really need to know where the animal was. But the algorithm in the background uses all the information it can get to try and locate locate where the animals were. You don't need to know it. This all happens in the background, but the more information you give it, the better your estimate is going to be in terms of bias and precision. So much smaller confidence intervals around your abundance estimate and much smaller bias in any given estimate. So then you put a bunch of sites through your study area, and then you record over some period of time. And you make your detections, and then from that you build your capture history. Those of you who work with mark-recapture data sets, it looks probably familiar, but in this case, we have, you know, four different calls. We have 10 different sites. At each site, there's three recorders, and then you have the ones for the recorders that detected a call, and for those detections for those records where you have a 1, you also get an angle when you use our recorders, our TREX recorders. Of course, this requires that first you need to analyze your recordings. You need to make you need to find the calls in your recordings, and those of you working with acoustic data know that if you do that manually, that is very tedious. So that's the work-intensive part about acoustic surveys. So you either do that manually and you look through all the spectrograms and listen to them, and then you find your calls there. I've done that for several hours of the data we collected in Laos. But we've also developed an automatic detector, which works by, well, you give it a training data set, and you train your detector, and eventually you can run your whole data set on your, you run the detector on your whole data set, and it comes up with probabilities that your call is in a short segment. So it cuts up your recordings into 3-second segments, and for each segment it tells you the probability that a female call occurred in that segment was somewhere between 0 and 1. And then it's up to you to find the threshold, where you get a good return of true positives. So something that the detector said it was a detection, that is actually a detection. So, but I'm getting very technical here. So we have the detector working quite well. That takes away a lot of the manual labor, but then the matching of the calls between the detectors, that is still the time-consuming part. We need to look at every call detection from, let's say, recorder 1, and all the call detections from recorder 2, and then you need to match them. Is that the same call? And for that, you have the information of the exact time when it occurred and the angle. And it turns out that looking at the spectrograms makes it a lot easier to make that decision, because some calls have 4 of these upsweeps. Sometimes there's 5. So that's one example. So in order to make this process easier, we've developed a Shiny app where you can click on a few call detections and it prints out the map, and it shows you the angles where they were calling from. So in this case, you see that this call here was probably not the same as these 2, but these 2 were probably the same call. That makes sense. So still a lot of development to do here, because as a biologist, of course, you want to see as little of that as possible. You want to plug in your recorders, push a button, and get your capture history back, or even maybe an abundance estimate. That's even better. So we're in the process of improving the algorithms that we have and making them as user-friendly as possible. because there's some urgency here because all 20 gibbon species are endangered to various extents. So it's a good thing to move this forward. Last, I have some technical information about why including angles is better, but I think maybe I skip these and you can just take my word for it. And with that, I would like to thank you guys for your attention.
Thank you so very much for sharing with us. We're going to open it up to questions, and we're going to bounce back and forth between questions online and questions in the room. And I can see we've got a question online. So go ahead, Roseanne.
Can you speak to incorporating information on whether a population has a random, even, or patchy distribution into your work, particularly when determining probability of detection?
Yes, I can say a few words about this. Let's go back to this slide. So far, I've spoken about what's called a design-based estimator for distance sampling data. And that was the part I skimmed over at the beginning, where you scale up from your density estimates at your lines to the density or abundance estimates at the lines, scaling up to the abundance estimate in your whole area by simply using a multiplier. How big is your surveyed area compared to the large area? But there's a completely different range of distance sampling models out there that take into account varying distributions. The design-based method works also, but it works only when you have a good survey design that is a good representative of your whole study area. But the what is it called? The model-based distance sampling has a different approach for scaling up. So here, what you would do is you cut up these individual lines. So instead of having one sample here, you cut it up into small segments. And the size of the segment depends on the size of your study area. So in small study areas, people have used two nautical miles, but in the ETP, I think Megan has done this, and she's used 9 or 10 nautical miles. And then you get a habitat covariates associated with each segment. So for example, depth, sea surface temperature, anything that you can think of that might affect the distribution of the dolphins. And then you can you get your density estimate for each segment now, not just for each line, but for each segment, accounting for detection probability within that segment. And then you fit a habitat model across your whole study area and express your abundance or density throughout your study area as a function of your covariates, whichever are the driving factors: sea surface temperature, ocean depth, thermocline slope. Those could all be factors. And then you get a better idea about the distribution of your animals in your study area. Does that answer your question? Okay.
Great. Thank you very much. If that did not answer your question, go ahead and put an additional question in the chat and we'll circle back to it. How about folks in the room? Any questions for folks in the room?
Thanks, Cornelia. Great to see that, and nice memories. You spoke about, I'm always interested in these assumptions of these methods. And one of the basic ones you talked about early on was the assumption that all animals will be detected right on the line. The G sub 0 equals 1, or you call it F sub 0. And we know in marine systems studying marine mammals that that assumption is violated simply because the study animals are underwater a lot of the time. And I wonder if you could speak a little bit to um, advances in addressing that assumption with, well, not only all marine mammals, but particularly with deep divers that can stay under for an hour or more at a time?
Yeah, thank you for that question. It's a difficult one. So I think initially for the when you design your distance sampling survey, you need to think about that assumption. Will you detect all the animals, or is there a reason to believe not? So, for example, with dolphins, considering that the observers can see 6 or 7 nautical miles ahead, how long does a ship take to cover that distance? For simplicity, let's say it takes an hour. It takes a little bit less than an hour, but for simplicity, say it takes an hour. And then if you think about that, the dolphins surface every 20 minutes, 10, 15 minutes, 20 minutes, whatever, you realize you have multiple chances of seeing it. And I think that was the way it used to be thought of, and that was considered good enough. And I wouldn't have a problem with it, but then Jay's paper crept up. So that's one consideration. Then of course with the deep divers, an animal that dives for 60 minutes, 90 minutes, or whatever, I think then there was a simple multiplier you could incorporate into distance sampling surveys where you considered also again that time it takes you to cross from here to the horizon. So like how long do you have to detect that animal, and how long in comparison is the dive cycle? So it was also a simple multiplier. And I know there was some papers by Jeff Lake and David Borchers, and they kind of improved the multipliers over time. That is as much as I know about the deep diving. In terms of dolphins running away, not the same issue, but of course you need to catch them before they run away. So that's a consideration there. Also, a related issue is that you need to set up your trials. So whatever I call the first platform making observations. So for example, the drone sets up the trials for the flying bridge, for the binomial mark-recapture model. So the drone needs to set up the trials that make sense. So that means all the trials need to be a random sample from the whole population. So for example, if they were too close together and the dolphins had run away, you'll only get trials for the dolphins that stayed, and the dolphins that ran away will never be considered. So that's a consideration. Make sure that the trials are covering the whole population. Sorry, I hope that wasn't too technical.
Okay, great. I think we have another question online. Go ahead, Roseanne.
Yeah, this is a request. Could you replay the gibbon sounds through your voice microphone, or maybe raise the volume so the folks online could hear them better?
Okay.
All right, everyone, that's as loud as it goes. We'll try. Oh, I know what it is. Hang on one second. Does anybody else have a question? What we need to do is I need to stop share for a sec and reshare where we have sound that we can share.
Okay.
So hang on one sec. If anybody has a question in the interim, let me know. Roseanne, anything else?
How difficult would it be to use hydrophones to increase the accuracy of detection in marine mammals?
You wouldn't, I mean, detection, I think the question was rather accuracy of abundance estimates rather than detections.
Accuracy of detections, population estimates.
Yeah. So usually the more detections you have, the better your abundance estimate gets. So with acoustic detections, you have a chance to make a lot more detections with a lot less effort. So for example, if you mount some hydrophones on the sea floor, you can leave them out for a duration of time as long as the batteries last and they keep recording, and then you retrieve them and you get a lot of detections over maybe the course of weeks or months. Whereas if you do visual detections, you're limited, you're very limited in terms of effort. So you usually survey the whole area. I mean, the part of the area that you survey, you only do that once at one specific time point. So you don't get a sample over time. So you get a lot more detections with acoustic than visual. So you have an increased sample size, but then to turn that into, that's the benefit. That's a big benefit. The downside is that you will get an abundance estimate of calls, and to convert that into an abundance estimate of animals, you need to apply a multiplier. How many calls does an animal make on average over X amount of time? So that decreases the precision again. So in the gibbon world, it makes complete sense to use acoustics, in my opinion, because I mean, I had zero chance of seeing a gibbon in the forest, but I'm the clumsy person who had no experience there. But even the people who go in the field there all the time, they might have not necessarily seen one before in the wild, but they hear them a lot. So that makes perfect sense. In the marine world, I guess it's a case by case thing. But people are moving more and more to acoustics. I think because it is cheaper to collect the data. I don't have the exact answer for you there, but in some cases it makes sense. It is an improvement, definitely. Yeah, but I don't think we're quite ready to move just to acoustics. So if you want to build an existing visual data sets, for example, you need conversion factors. How do I convert an acoustic estimate into a visual? How do they compare? So that's another question that needs to be addressed then.
All right, I think we can play this now and folks online can hear it if you. Folks online, let us know if you can hear now.
was it.
All right. I think that we are at time, and I know that there might be a few more questions, but I wanted to give us a chance to say thank you very much for our speaker for today. Wonderful, everybody. Thank you so much for being here. I hope to see you next week or at the Big Blue Film Festival this weekend. All right, everyone is invited to go to Beer One after if you would like to continue this conversation or if you have questions. So that sounds all like a wonderful idea to keep everything going. So hopefully we'll see you there. Thanks, everyone.